SARS-CoV-2, the virus that causes COVID-19, is currently keeping the whole world in suspense. What is known today is that a lot of people undergo a SARS-CoV-2 infection, but show only minor symptoms or no symptoms at all. Therefore, a high number of unreported cases is assumed. The question arises, is there a way to know whether someone has already been infected? Yes, with an antibody test. Our immune system has two strategies for dealing with invading pathogens like SARS-CoV-2. The innate immune system is available from birth. The acquired immune system develops tailor-made weapons against each enemy, specialized antibodies. At the beginning of an infection, there are only a few antibodies. If the infection progresses further, more and more are produced. They bind to the viruses and do not let go. In this way, the viruses are marked and can be recognized, inactivated, and finally rendered harmless by specialized white blood cells. This process usually continues until the infection stops in most infected people. Therefore, even infected people without symptoms have antibodies against SARS-CoV-2 in their blood. Antibody tests measure the immune response to SARS-CoV-2 in the blood. In technical jargon, antibodies are also called immunoglobulins. There are a total of 5 different immunoglobulin classes. 2 of them are particularly relevant for an antibody assay. They are called immunoglobulin M and G. IgM is one of the quick starters. This antibody is produced for the initial fight against SARS-CoV-2. In the second major wave of defense, IgG plays the leading role. Typically, IgG can be detected in the body for a relatively long time. Total Ig tests that predominantly detect IgM and IgG are therefore valuable for the detection of a past infection with SARS-CoV-2 and COVID-19 disease. To detect antibodies in the blood, there are several testing techniques available. One is by so-called electrochemiluminescence. In the lab, proteins that match those of SARS-CoV-2 are produced in large quantities using molecular biological methods. They are divided into 2 parts. One portion is labeled with a chemical element, ruthenium. It plays the role of a biological flashlight. The remaining part of the produced proteins are labeled with biotin, which will act like an immunological anchor. These 2 portions are then incubated with the patient's blood sample. If there are antibodies against SARS-CoV-2, the 2 differently labeled proteins act similarly to tweezers, targeting the antibody, so to speak. The result is a double antigen sandwich complex. In the next reaction step, tiny paramagnetic beads bind to the biotin anchor. These complexes enter the measuring cell of the analyzer. A current is applied to an electrode, turning it magnetic. All beads remain on the surface of the electrode. A special solution is added, oxidizing the ruthenium. This turns the biological flashlight on, indicating that there are antibodies in the sample. It is still not known for sure whether people are really immune after having been infected with SARS-CoV-2 and recovering from COVID-19, and how long this acquired immunity may last. Intensive scientific studies are underway to find out. With an antibody test, health authorities could certainly determine the exposure status of the population. The results could help make important decisions about public health measures, such as the reopening of public facilities. If further research confirms that recovering from COVID-19 signifies immunity, hospitals could potentially use this test to help allocate recovered healthcare workers to care for patients with COVID-19. And should a vaccine be available soon? An antibody test can also help identify the people who would need the vaccination most urgently.